I'm really happy to introduce Hans-Jörg Rheinberger. Most of you probably know who he is, but maybe some don't. Hans-Jörg Rheinberger is one of the former directors of the Max Planck Institute for the History of Science in Berlin. And he's also quite interdisciplinary. So he's done philosophy, but also biology. He actually did quite some things in molecular biology before coming back to philosophy and to the history and philosophy of science. He published largely on experimental systems, on epistemic things, and his latest book is called Split and Splice, a phenomenology of experimentation, that appeared in 2023. And I'm particularly happy to have him here because actually without him in a certain way I wouldn't be here, because I would have never met Didier, and then I would never have moved to Brussels, because he actually employed both of us, Didier as a postdoctoral researcher and me as a PhD student, quite along almost 20 years ago actually. So I'm very happy the floor is yours.
Yeah, thank you very much, and thank you for inviting me to this meeting, although I'm not very sure that I can contribute a lot to Latourian scholarship, at least not as it is done today. Might be a different thing if one goes back to the 1980s. Quite obviously Latour was somebody else, yeah, and we also, I think, should not forget that there is a history behind people and what they write. But at least this meeting gave me the opportunity to reread the modes of existence, although I then in the end decided not to start with an emulation of Latour, but to do something different, namely to try to contribute to a better understanding of the historical dynamics of scientific practice. And why I am doing that, I think it could at least be an effort to test how far the terminology that Bruno Latour proposes in modes of existence will be able to take us, because I think that is not to be taken for granted from the very outset. Now, my point of departure is that there is not a single trajectory or chain of reference, for that matter, that can do justice to the winding paths of scientific development, and particularly not of scientific development today. According to the perspective chosen, different lines of action and trajectories of entities can be discussed and also can become visible. And it is the ensemble in the end that characterizes the development of a scientific field. Among them are trajectories of experimental systems. I have worked a lot about that. Migrations of epistemic things. Pathways of technical objects. Movement patterns of experimental cultures. Organismic migrations, and the latter, at least as far as the life sciences are concerned. And I think all of that has to be considered if one would like really to get a full picture of what scientific practice is all about. So, might be might be a summary, but it does not tell us everything. At the end, I hope at least we will be then able in a better position to delineate how this multiplicity relates to Bruno Latour's mode of existence that he accords to the sciences. Now, to begin with, let me recall a remarkable, as I see it, a remarkable passage in the classic of Ludwig Fleck, his Genesis and Development of a Scientific Fact, a book written in 1934, published in 1934. I will quote this passage in full length. I quote, "It is very difficult, if not impossible, to give an accurate historical account of a scientific field. Many developing strands of thought intersect and interact with one another. All of these would have to be represented, first as continuous lines of development, and second, in every one of their mutual connections. Third, the main direction of the development, taken as an idealized average, would have to be drawn separately and at the same time." End of quote. Now let me now briefly present 5 kinds of such Fleckian strands or sequences that can be specified in the empirical sciences and that importantly constitute an equal number of temporal regimes with their own traits, each of them, and characteristics, and the resulting frictions also between them. And I think the notion of friction is important here, because you cannot think of any development as being without frictions. Now, first, trajectories of experimental systems. Now, I have discussed them at length in my book. Some of you might know it, Toward the History of Epistemic Things. And so I will not go into the details here. But as a rule, such systems stretch over periods of time that often, interestingly, coincide with the productivity cycle of a particular research group, or even an individual, that is something between 10 and 20 years. Experimental systems can be seen as loose articulations of materials, of research technologies and procedures, whose couplings are, however, sufficiently strong to shape a research process around a particular research object in a way that promises to discover something new about the target of research. That's what research is all about. Experimental systems are temporally limited, thus, and at the same time locally constrained research environments. As such, they are endowed with an underlying material continuity. Along such an axis of continuity, however, they change permanently, at times incrementally, at times also disruptively at the conceptual level. So, and we have to distinguish these two and to look at their interactions. It is, however, always possible that such systems come to a sudden end, particularly if an alternative system promises to advance the same problem complex in a more productive manner. The history of the sciences is replete with such abandoned systems. To give just one example, in the late 1950s, 3 different experimental systems were positioned in the ambit of a burning problem whose solution then appeared imminent, but not yet tractable: the cracking of the genetic code. Sidney Brenner and Francis Crick's system in Cambridge was centered around the analysis of nucleic acid of bacteriophages. Heinz-Günther Wittmann's system in Tübingen concentrated on the analysis of mutants of tobacco mosaic virus, and the system by Heinrich Mattei and Marshall Nierenberg in Bethesda aimed at an analysis of bacterial protein synthesis. Although the latter had not been established from the outset with the genetic code in mind, that was not the kind of vision that people had when they established that system. In the early 1960s, it came to render the other two systems obsolete within a time span of a few months. This was essentially due to the temporal characteristics of the in vitro protein synthesis system. It allowed for carrying out a decisively denser succession of experiments and a much more direct interpretation of the results. It effected a condensation of the experimental work of several orders of magnitude, and so it gained upper hand. And the other two groups that had invested equal amounts of time, about 10 years each of them, had to look for something else. Now I come to my second point, migrations of epistemic things. So I talked about systems, now about things. The example I just gave you plainly shows that experimental system and epistemic target have to be distinguished from one another. The epistemic objects are the materially and theoretically underdetermined entities on which the scientific interest of those who run an experimental system focuses. The latter, in turn, the system, consists of the research technologies and procedures that help to give contours to the things of epistemic interest. In a concrete research environment, the investigated phenomenon and the research technology are specifically correlated. They condition each other. And yet, materially, they can be of a very different nature. An enzyme and an ultracentrifuge, for instance. However, there will be no knowledge effects without an epistemically productive interface between phenomenon and technology. An instrument that cannot be adjusted to an epistemic object has no scientific value. And an epistemic object that cannot be made compliant with the instrument in one way or another is simply not a phenomenon that can be investigated under scientific auspices. As simple as that. The concept of the epistemic designates the object of knowledge in its essentially underdetermined form, a circumstance that makes it relevant for research in the first place. It is exactly for that reason that I speak of epistemic things. The notion of thing connotates the indeterminacy that lies at the core of research objects. And the etymology of the word refers to the circumstance that we are concerned with a thing that is put on trial. I think Latour has also written about that. The concept or category, as Latour possibly would say, of the technical, in contrast, is associated with specified contours. It is not up for debate, at least not for the moment being. It has the character of a closure. The epistemic and the technical remain mutually dependent and draw their respective specificity from just that relation. Now, as already mentioned, the material continuity of an experimental system does not at all imply the continuity of the epistemic things dealt with in its context. On the contrary, it is a prerequisite for conceptual change at certain turning points in the development of an experimental system. Let me give an example again. In 1894, the botanist Karl Correns began to cross corn varieties. Shortly thereafter, his work branched out to pea varieties. And all these details are important here, in the hope of experimentally producing a biological phenomenon that at that time was called xenia. Xenion. That is the direct influence of the pollen on the mother plant, in order to be able to investigate its physiology more closely. So that was the idea behind setting up the system, a hybridization system in the event. In this, Correns was successful with his mice varieties, although not with his peas, where he was struck instead by the regularity with which, in the second experimental generation, the characters of that parent part that had vanished in the first crossing generation reappeared. Following this new track required only comparatively small modifications. On the whole, Correns could continue his experimental regime and achieve statistically robust numbers about the progeny ratios of his crosses. Consequently, the search for traits on the paternal plant in the seeds and fruits of the mother plant, that was the beginning of all that, was replaced, completely replaced, by the experimental and theoretical corollaries of Mendel's rules that were actually rediscovered just at that point. The new epistemic things for Correns were the Anlagen, as he called it, not yet genes, that were apparently inherited independently of each other. Under the label of genes, we know that all, they would soon begin their trajectory through the biological experimental systems of genetics that would extend throughout the 20th century. Now, there is another interesting point here that probably we should not forget, and it is also a point that might have contributed at least to this ongoing debate about why Mendel's work was not taken up earlier by other people in the field. He had published a paper already in the middle of the 19th century, and it was taken up only at the end. I think one of the core things is that with that, a change in the vision of what an organism is looking like and composed of and working, and how the its genetic material is being transmitted from one generation to the other. What is at the core of all? Chance. Yeah? So it's a chance distribution. So that organisms should rely on pure chance. It's hard to to take up in a way. And maybe that is one of the things that is behind this delayed reception of Mendel's work. No, but once again, we see here that the example shows that the trajectory of epistemic things is not identical with the trajectory of experimental systems. Not only because the former can be displaced from an existing system and replaced by a different epistemic object, but also because they can jump from one experimental system to another. Let us stay for a moment with the example of the genes, one of the cardinal epistemic things in the life sciences of the 20th century. Experimental hybridization systems dominated the first 4 decades of the century. The emergence of molecular biology led to the addition of biophysical systems such as X-ray structure analysis of biological macromolecules and the mutagenesis of viruses induced by X-rays, as well as biochemical in vitro systems like the above-mentioned protein synthesis system, to mention just a few. And finally, the emergence of gene technology from the 1970s onward led to the manipulation of genes and led back actually to the manipulation of genes in living cells by means of molecular techniques. Techniques that in turn resulted from the transformation of molecular epistemic things into molecular technical tools. So we have also this conversion here between the epistemic and the technical, ongoing and in both directions. Now, what I would like to take from this discussion is that epistemic things can migrate from one experimental context to another. These migrations are not linear and not directed, but they are ramified, multiple, and intermeshing. On the one hand, they promote the stabilization of certain aspects of the epistemic objects in question. Thus, in contexts, again an example, in contexts of today's biotechnological production, precision-tailored genes function as molecular replication machines. On the other hand, they do not come to a rest as epistemic objects, because the multiplication of their uses as tools is accompanied by the emergence of new blind spots. Through migrations of this kind, the experimental systems involved become interconnected to a certain extent, resulting in multiply networked experimental webs that are mutually associated through materially mediated pathways. In this way, and in the context of such experimental cultures, experimental systems only marginally related, because differing in terms of the relevant technical components, nevertheless come to communicate with each other. Now, my third point, pathways of research technologies. The technical components of experimental systems, that is, the research technologies and other standardized and custom-made parts of such systems, exhibit characteristic migratory movements and trajectories as well. In contrast to the history and migratory movements of experimental systems and epistemic objects, there is an extensive, that is an interesting observation, by the way, there is an extensive literature on the history and dissemination of research technologies. So there is no need, I think, to focus here on this point in detail. I think every major research technology in molecular biology has gotten its own book-length study. Now, what is of interest here are the temporal shapes, the movements in time that research technologies perform in the space of the epistemic. We can roughly distinguish two phases here. In the phase of the emergence of a new research technology, they mostly crystallize by learning to handle and manipulate the materials, procedures, and energy forms that determine their core structure. Think of a microscope, for instance. There is often previously unimagined effects of the investigated material that open perspectives on for a new technology. And that happened, for example, in the case of electron microscopy. Then, second phase, in the ensuing development, instrument and object of investigation enter into a tight reciprocal interaction with each other. For electron microscopy, this becomes particularly evident when we look at its uses for the visualization of organic materials. This mutual instruction can trigger epistemic as well as technical effects. It is not unusual, therefore, that research technologies themselves, and again, electron microscopy is a good example, will move over longer time spans in an experimental, essentially epistemic realm, and can even assume the status of something like a technical experimental system of its own. Sooner or later, however, and this is the third phase, most research technologies become encased in more or less closed packages. For their users, that transforms them into black boxes. They are no longer tinkered upon in the laboratory. Instead, they are industrially produced. Only the insertion into a system in question remains open for limited experimental adaptations. And you can also see that that restricts certain options and possibilities for doing research. On the other hand, however, their horizon of application is widened. Their migration is facilitated, and they become integrated into a much greater horizon of experimental environments. Something comparable to black boxing can also happen to experimental systems as a whole. They can be transferred into what is usually called a test system. As standardized subroutines, they become part of other experimental setups. This shows that although all these components of the experimental process can and must be differentiated, they are by no means locked into these roles forever. Rather, they can change places in a dynamic manner. Only concrete historical analysis can convincingly show what function a particular element fulfills in a given experimental context. So there is no set of rules from which you can dedicate this kind of thing. Now I come to the fourth point, fourth kind of movement, on the motion patterns of experimental cultures. The migration phenomena mentioned, whether related to epistemic objects or to research technologies, decisively contribute to the constitution of what can be called cultures of experimentation. They let them appear as multiply reticulated ensembles of experimental trajectories of the different kinds of components that I have already described. In such compound experimental structures, fusions or hybridizations of experimental systems that originally had developed separately, even widely apart from each other, can happen. They can come together, even fuse. It can also happen that having become too rich in options, experimental systems split up. With an experimental culture, such bifurcations or even ramifications can lead to niches in which the exchange between the systems involved is particularly intense. Generally speaking, cultures are looked at as collective ensembles that rely on something their members share or what circulates in them. Might even be a general definition of what the culture is all about. Yeah? It's about sharing, actually, and it's about circulation. Cultures of experimentation, in particular, are characterized by a threefold circulation. First, a circulation of materials to which their epistemic objects relate. Second, a circulation of research technologies that can be used in different experimental setups. And third, the circulation of know-how that researchers carry with them when they move or change places within such a culture, which frequently is the case. Certainly in the course of an education of somebody who wants to become a scientist, who has a phase where he jumps from one laboratory to another for about 10 or 15 years. Now I come to my point number 5, organismic migrations. Might faintly relate to what Chris had to say this morning, but on a much more restricted, at a much more restricted level. So let me conclude the broad range of these temporal and spatial patterns with this particular case. It is neither an organism is neither an experimental system, nor an experimental culture, neither an epistemic nor an exclusively technical object. We could perhaps capture it with the concept of strategic research material, which we owe to Robert Merton, actually, the forgotten sociologist of science of the 1960s and 1970s. Merton used it to designate materials that for certain reasons are particularly suitable for the study of natural phenomena, that show themselves here in a succinct form or can be made accessible in a particularly obvious way. Such materials often have a long history that does not necessarily have to be situated in the space of the epistemic to begin with. Not at all. They can, however, end up in science on their way through completely different cultural contexts and disappear from it as well again. In the biological sciences, this often concerns certain organisms, as I said. Today, they are referred to as model organisms. In a very special way, model organisms are carriers of the regimes that are essentially determined by their reproductive cycles. Rep steps in. In the history of biology, from the late 19th century onward, a tendency can be observed toward the economization of time, and with that, not only the life sciences, and with that, the transition to model organisms that reproduce ever more quickly. Early classical genetics still worked with plants that were bound to an annual reproduction rhythm. With insects, in particular the fruit fly Drosophila, model organisms were introduced into the laboratory whose reproduction cycles were reduced to months or even weeks. Early molecular biology finally specialized in unicellular organisms such as yeast and bacteria with a reproduction cycle of hours or even less under optimum conditions. The impact of these organism-specific time regimes on the rhythmicity of experimental systems in the life sciences can hardly be overestimated. Now, the model organism to which I would like to focus here as an example is a very strange animal. Certainly nobody of you will know it. With a medium range reproduction cycle, the flower moth Epistemic cuniella. It is an unspectacular insect from the family of pyralid moths, something for the biologists among us. Assumed to be native to Central America, it found its way to Europe in the 19th century in the wake of colonial traffic. The precise time of its arrival on the European continent is unknown. Here in Europe, it first attracted attention as an unwanted guest of grain mills, where under Central European weather conditions it bred in 3 generations around the year. Consequently, the moth began its scientific career as a pest insect, an object of investigation of applied entomology, with the aim to control its spread or even to eradicate it. In its role as a harmful insect, the moth first fell into the hands of agricultural scientist and phytopathologist Julius Kühn, therefore its name, Epistemic cuniella. Kühn had been conducting research and teaching at the University of Halle in Germany since 1862. In the summer of 1877, he collected several specimens of the moth from a mill that processed American grain and sent them to the entomologist Philipp Christoph Zeller in Stettin for determination. Zeller was a specialist in smaller moths and a long-time secretary of the Entomological Society of Stettin, a tiny town in the northern part of Germany. So it becomes very local, the story here. And it is everything else than local in the end. His description of the insect under the scientific name Epistemic cuniella appeared in 1879. Now the flower moths received increased attention during the First World War. It accumulated in the big mills that had become centralized in the course of the war for food supply reasons and propagated there en masse. The parasitologist Albrecht Haase, who specialized in insecticides, but also in biological warfare agents, was the first to explore their mass culture as a laboratory insect in the early 1920s. So he tried to cultivate the insect itself en masse in order to learn more and to have enough material to work with at the Biologische Reichsanstalt in Berlin, Dahlem, around the corner of the Max Planck Institute for the History of Science. The building still exists. Above all, Haase was interested in the biological battle against the flower moth using another insect, namely the wasp Habrobrachon juglandis, which deposits its eggs in the caterpillars of Ephestia and thus kills it. In the later course of the 1920s, in turn, the Göttingen zoologist Alfred Kühn has nothing to do with Julius Kühn. They are completely unrelated. Adopted the mass cultivation procedure that Haase had developed at that time in his zoological institute in Göttingen. At that time, Kühn was interested in, among quite a number of other things, in the variations of that wasp that constituted the dissertation subject of one of his students, thus not on Ephestia, but on the wasp that led its eggs into Ephestia. Thus, initially, his group used the flower moth Ephestia as a feed animal for another animal. Presumably by chance, they became aware of conspicuous wing mutations in the moth, not in Juglandis habrobrachon. As a consequence, Alfred Kühn embarked on a study on the genetic, physiological, and developmental aspects of wing coloration and wing patterns of Ephestia. But this long-term endeavor, he had envisaged to pursue it over quite a number of years, in the course of which Kühn and his co-workers, within 4 or 5 years, they had inspected more than 100,000 of these animals. And the project failed to bring the success they had expected. Basically, nothing came out of it. 2 publications. The situation changed when 5 years into this project, a technical assistant of Kühn detected a red-eyed mutant of the moth in their mass cultures. It soon became clear that the inheritance of the red eye color followed a recessive Mendelian distribution. The mutant, so it appeared, lacked a substance that obviously circulated freely in the body of the wild type, as Kühn's co-worker Ernst Kaspari was able to show in a series of transplantation experiments between mutated animals and wild type animals. With that move, Ephestia kühniella advanced to the main model organism of emerging physiological and developmental genetics. A step that would become obvious only about slightly less than 10 years later in its importance. It would be another 10 years, however, before the substance under observation was characterized biochemically. It turned out that it was an intermediate product in the composition of the eye pigment. The substance could not be formed if, as the result of a gene defect, the enzyme that catalyzed this intermediate step was missing. With that, all the ingredients of biochemical genetics were identified. Gene action chains consisting of genes and gene products, that is, enzymes, as well as metabolic chains whose intermediate steps were catalyzed by the enzymes and not catalyzed when the enzyme was missing. However, next problem, the problem was that the experimental system built up around Epistia, while allowing access to this particular metabolic chain, did not permit an assessment of the genes and their products. The genome of the moth with its 30 or so chromosomes was much too complicated to achieve this at that time. No chance to do that. Thus, the life of the flower moth as a genetic physiological model organism remained restricted to a narrow research window between the two world wars. Fungi such as Neurospora crassa, bacteria such as Escherichia coli, and viruses such as tobacco mosaic virus, as well as bacteriophages, would soon replace it as the new model organisms of the burgeoning fields of molecular biology and molecular genetics. And the work of Kuhn was largely forgotten. Almost nobody knows him today anymore. Now, the scientific existence of the flower moth thus formed a chain, with its starting point around the middle of the 19th century and its end point around the mid-20th century. Maybe there is even a precursor to it in Central America. This would have to be found out. As an object of knowledge, it began its career as an unwanted colonial import that made an impact as a food pest. It advanced to become a taxonomic unit in entomology, gained new life as an experimental organism in biological insect control, and finally mutated into a model organism in the epistemic space of emerging physiological genetics. Then the moth as a model organism disappeared from the scene of knowledge altogether. The French physiologist Claude Bernard once remarked in his notes that the epistemic chains of discoveries in physiology would constitute a historical connection, but that the links of the chain could by no means be derived from each other. Biological necessity. The same can be said, and rightly so, I think, from the contingent steps of the scientific career of the flower moth. Yeah, so that is more or less also basically the end of of this case, which I find as such a very interesting and a very telling one. But maybe I conclude with a few reflections is too much, so it is not even reflections, it's actually rather questions I have. Because in preparation for this workshop, I have read Bruno Latour's inquiry into modes of existence, again from front to back. And I must confess that I was not able to make up my mind conclusively about that giant enterprise. Do we have to do with a mild and elastic version of Luhmannian systems, theoretical thinking? Do we have to do with a critique of the ideology of modernity, a subreptive reintroduction thus of what Karl Marx called the critique of false consciousness? Because Latour's moderns have false conscious about what they are doing themselves all over the place. They simply do not see each other. Will Latour's fear come true that he expressed actually himself at the end of the book with the following words, I quote: "Specialists will have no problem to shred this fragile assemblage. It will then never have been more than yet another schism, another grand narrative." End of quote. And I think it is not by chance that he put this down. Or do we have to see in it the announcement of a new epoch in history? So when I heard to you talking about them, maybe more or less all of them are pointing to that latter option and interpretations. But I have questions to that and no solution. Thank you for listening.
Thank you very much, Hans-Jörg. Is there anyone who wants to jump in directly and has a question or a remark? John?
Yeah, I'll try. Thank you very much, Hans-Jörg. Fascinating recapitulation of different ways of looking at experimental systems, and in a way very aligned, I think, with Latour in the sense of multiple passages and trajectories that intersect and cross in an unexpected way. So you focused on experimental sciences, particularly biological, and these 5 dimensions of them. And that was a fascinating reconstruction. But of course, I want to jump to your open-ended conclusion and ask how this very robust picture of scientific practice relates not so much or not only to Latour's take on breath, which we already heard this morning. Many people who work in life sciences find insufficient. Also, breath seems insufficient to many people who work in life sciences. But more generally, how this proposal or this version of experimental science that you've shared interacts with the bigger picture of Latour's project, which is to ask, as he always has, how does science relate to society? To go very quick. How does it shape society? How does it come from society? And one way to sharpen that question, so what do you think about where you stand in relation to that question and where Latour stands? Yeah. And one specification I make is that your work is remarkable in relation to traditional philosophy of science, in that it seems to be very unconcerned with demarcation. And early Latour also didn't care, or was very, very clear about saying demarcation is a false question. Science and other things are constantly mixing. But what's striking about the modes of existence is that he is returning to demarcation. He wants to say, this is what makes something science, just as this is what makes something a different mode, and different from other things. So, both, where do you stand in relation to science in relation to society, and also, where do you think you now are in relation to the question of demarcation?
Yeah, I think this is probably this has been the main point of concern for me when reading the modes of existence again, that it appears, at least, maybe if one goes into more detail of each of these modes, one will be able to open them up. But in a general description, such as in 25 pages in one of the chapters of the book, they appear to close upon themselves, yeah? And to have their own, each of them has their own truth regime, and it is always decided from within that truth regime what belongs to it and what is excluded. And what I would like to show is that these trajectories are, at every step they are going through, are open, and they are deflected into different, can be deflected into completely different directions. And the example that I was giving at the end of this of the moth, yeah, of Ephistia cuniella, I think it shows at many points where the rest of society comes in, yeah, because you have colonial traffic, you have the import of American grain into Europe because of food shortage and all that, the political things related to that, and then you have, of course, the war effort and the usage of biological entities, not only as being able to fight biologically against invaders and other things, but also to use them, to dual use them, to use them for another purpose. And all of that, at each and any point of this trajectory, you can plainly see how the bigger picture intrudes into this trajectory, be it politics, be it economy, be it war, or whatever. So I think, at least for me, it appeared to be a striking example, and not at all confined to the laboratory as we are usually thinking about these matters. You know, there is a closed laboratory and what is happening there.
Yes, I wanted to follow along the same line about the centrality of experimentalism within modern science.
Yeah.
And I have the feeling that in the Latour's proposal, it's one of the possible scientific practices, and more, obviously, laboratory is associated with Latour. The first Latour probably, but more and more the field work, the terrain, became more and more relevant until the end with the territorial approach. And so I feel a tension, in fact, also in the way of dealing with scientific practices. So I would like to have your view also on this form of pan-experimentalism that occurred during the 20th century, the multiplication of experimentalism.
Yeah, so I have written a little bit about that in my last book. So, of course, we, in a way, if we would overgeneralize things, we could say in the 20th century our societies have entered as a whole into an experimental phase, into an experimental status. Nothing is anymore fixed or appears to be fixed forever. And everything has to be tried out. And again, one little point to come back to John's question. The other way around it works as well. So science intrudes into economy, and you can see that in the development of molecular biology that led to gene technology, and today is one of the major players on the market as a whole, with its products and its inventions.
There are two more questions. Maybe Kim, we can assemble them so we don't get too late. So it's Kim and Chris.
I want to go back very shortly to your final questions. Could it be that one of the differences you're actually pointing at here is something that you perhaps want to emphasize as a historian, is that if we think in terms of modes of existence, then there is some kind of, we tend to think in terms of conditions of felicity and some form of necessity to demarcate the different modes, whereas you as a historian would like to stress the historical contingency of scientific progress. And I think this is a sort of tension we, at least for myself, I also tend to grapple with when thinking both in terms of Latour's theory and in more historical approaches to scientific technology. We should also follow, but at some point there is this slight takeoff perhaps from historical contingency. And perhaps one question is also, what does historical contingency mean? Is anything possible, any kind of combination of movements?
There was
I just quickly want to point out that one of the interesting overlaps here is Charles Elton again, and in that book that he wrote, The Biology of Invasions, there are many moths, sadly not Epistemic, but many moths appear in the same region alongside rats and other kinds of things that invade. But for the purpose of non-experimental science, for in his case, ecological modeling. And so I guess if there's a question, it's a little bit how to account for the modulation of epistemic objects from experimental settings to non-experimental settings when it gets picked up. So Elton would be a great case to think with there. There might be other places in pest control because there's so much observational work where you can't experiment on wild populations. And so.
Yeah, I would be happy if I would be able to answer that question. With respect to contingency, yes, I think if people who are really interested in history will have to deal with contingency anyway, so there is no way out. As an anecdote, it comes to my mind again, and often again, when I met Bruno Latour, mostly so in the late 1990s and the early 2000s. He always, when we met and started to discuss, he always stressed, I am not a historian of science. I am not a historian of science. And I think a little bit of a historical perspective is also missing in the modes of existence.
I don't know, to me, but that's only my perspective. I do get that there is tension, but I have the impression one could also very much read your paper as a kind of zooming into one, maybe not one of the modes, but maybe
certainly.
But then we learn, and I think Latour would have been very interested by this, that we
to
actually
explode it from within.
Yeah, and also that you need, for example, two different versions of rep. That is rep more in terms of identical reproduction, that might be rep, and rep more as a metamorphosic thing, so rep met or something like this, in order to produce reference in certain fields of research, like in genetics, for example. I don't think there is really a total contradiction or an opposition. I have the impression that what you did was exactly what we asked you for. If you have the impression that there is an opposition, that is to show that things are maybe much more complex than what can be done in one book that wants to sort of deal with the entirety of modernity.
Let me add one thing at the end. I just wanted to make it as a remark, and I myself have to think it over again and also go back to Bruno Latour's book concerning the term metamorphosis. Because if you take this as a biological entity, and the idea of metamorphosis comes out of biology, it is a kind of development without change, because if an insect, for instance, metamorphoses from an egg into a caterpillar and finally into an imago and back again, it ends up in a circle. And so but I have to go back myself and look at whether this is in the background of Bruno Latour or not. Yeah, okay.
On this note, thank you very much.